Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome to the deep dive. Today, we're unpacking some really cutting-edge research. We're talking about malaria. And, it often feels like maybe it's a problem of the past, but it's absolutely a devastating issue right now.
It really is. The numbers are quite sobering.
Yeah. Just think about this. In 2023 alone, malaria tragically claimed around 600,000 lives globally.
600,000. It's staggering.
It is. And despite, decades of huge global efforts, insecticide-treated bed nets, spraying indoors, anti-malarial drugs, the progress we've made is, it's actually starting to erode.
That's the worrying part. We're seeing increasing resistance, both the mosquitoes becoming resistant to insecticides and the parasites themselves evolving to resist the drugs we rely on. It's this constant evolutionary arms race.
An arms race. That's a good way to put it. And it feels like we've had a point where we urgently need something new, something genuinely innovative to turn things around.
Exactly. We need new strategies. And that's why this recent study is so interesting.
And that's precisely our mission for this deep dive. We're going to unpack a really groundbreaking scientific breakthrough. It was just published in Nature, a top journal. This research introduces a totally novel genetic approach. It could seriously help malaria elimination efforts by focusing right on the mosquito. But, and this is key, it has a surprising twist that sort of tackles head-on some of the big worries people have had about genetic interventions.
That's right. It addresses those concerns quite elegantly, actually.
So get ready, because we're going to explore how one tiny genetic change, just a single tweak, might make a massive difference.
Okay, so let's get into the specifics of this study. As you said, the rising resistance is creating this urgent need for new ways to control malaria.
Yeah.
Now, the idea of genetically engineering mosquitoes, it isn't brand new. People have explored this before.
Right. I remember hearing about other attempts, but they often ran into problems, didn't they?
They did. Significant hurdles. Sometimes the genetic change didn't work that well, limited functionality, or, importantly, it imposed a fitness cost on the mosquitoes. Made them weaker, less fertile.
Which kind of defeats the purpose if they can't survive and spread the trait in the wild.
Precisely. And then there were those, bigger ethical and ecological concerns about potentially unintended consequences from making permanent genetic changes to wild populations.
Big concerns. So how does this new study approach things differently?
Okay, so the star player here is a specific mosquito protein. It's called fibrinogen-related protein 1. Let's just call it FREP1 maybe. Oh, wait, sorry, fibrinogen-related protein 1. We should stick to the full name.
Okay, fibrinogen-related protein 1. Got it.
What's really fascinating is its dual role. It's absolutely essential for the mosquito's normal life, its physiology, its survival.
Okay, so the mosquito needs it.
Yes. But critically, it also acts as a key facilitator, almost like a bottleneck, you could say, for the malaria parasites, the Plasmodium parasites, to get across the mosquito's midgut lining, the epithelium, and continue their life cycle. If that fibrinogen-related protein 1 isn't working right for the parasite, they really struggle to establish an infection.
So it's like a gatekeeper inside the mosquito that the parasite absolutely needs to get through.
Exactly, a critical gatekeeper.
But here's where I think the real genius comes in. Instead of just trying to, break that protein entirely, which could harm the mosquito.
Right, because it needs it too.
They focused on a naturally occurring variant of the gene for fibrinogen-related protein 1. So they found a version that already exists in nature.
That's the core innovation, yes. They found a subtle difference, just a single amino acid change in the protein sequence.
Wow.
Specifically at one position 224, the usual amino acid there is leucine. Let's call it L224. That version makes the mosquito susceptible.
Okay.
But there's a naturally occurring variant where that leucine is replaced by a different amino acid, glutamine. Let's call it Q2224. And this version, the Q224, actually makes the mosquito resistant to malaria.
Naturally resistant. So they didn't have to invent the resistance, just harness it.
Precisely. It's like finding a natural superpower already present in some mosquitoes and figuring out how to spread it.
That's incredible. One tiny change, L to Q, susceptible to resistant. So
yeah.
How did they actually engineer this? What was the method?
They used CRISPR-Cas9 gene editing. You've probably heard of it. It's like molecular scissors for making very precise changes to DNA, to deoxyribonucleic acid.
Yeah, Very precise editing.
So they used it to create these highly controlled strains of Anopheles stevensii mosquitoes. That's a major malaria carrier in Asia.
Yeah.
And these strains differed only in that one amino acid position. Either they had the susceptible L224 or the resistant Q224. This let them really isolate the effect of just that single change.
Very controlled experiment. But then how do you get that resistance to spread? They developed something called a linked allelic drive system. Sounds complex.
It is a bit technical, but it's crucial for making this practical. Basically, this system is designed to boost the inheritance of the protective Q2224 version of the gene.
Boost inheritance. How?
It uses a specific guide, a guide ribonucleic acid, or a guide RNA. Think of it like a tiny GPS guiding the CRISPR machinery. It targets the susceptible L224 version of the gene in the mosquito's reproductive cells, the germ cells.
Okay, in the eggs or sperm.
Exactly.
Uh-huh.
And it precisely converts that L224 into the resistant Q224. So when these mosquitoes reproduce, more of their offspring inherit the resistant Q224 trait than you'd expect by normal chance, by normal Mendelian genetics.
So it biases inheritance, pushes the resistance gene through the population faster.
That's the idea. A biased inheritance to accelerate the spread.
Now, this connects to what you mentioned earlier, the phantom solution. This sounds like it tackles those big concerns about permanent changes.
It does, and it's a really clever part of the design. Unlike some gene drive ideas that would theoretically spread and stay in the population forever.
Which makes some people nervous.
Understandably. This system can be engineered to be self-eliminating. Phantom. It spreads the resistance, but then it's designed to fade away over time.
How does it fade away?
They insert the genetic machinery for the drive system into a part of the mosquito's genome where having it long-term actually causes a slight disadvantage, a fitness cost.
Okay.
So the drive spreads the beneficial resistance trait initially, but because the drive machinery itself eventually becomes a burden, it naturally gets selected against and disappears from the population after, say, a number of generations.
So you get the benefit, the spread of resistance without the permanent genetic footprint. That seems like a major step forward in addressing concerns.
It's a very significant advantage, easing those critical concerns about permanently altering wild populations. A temporary intervention with potentially lasting impact on the disease.
Really pivotal. Okay, so they engineered this, they designed the drive. What happened when they tested it? What were the results?
The results were pretty remarkable. Mosquitoes carrying that resistant Q224 version showed really strong, robust resistance to malaria infection.
Okay, let's break that down. What about against the main human malaria parasite, Plasmodium falciparum?
Right, so they exposed the mosquitoes to infection levels, similar to what you might find in the field. Fairly low concentrations of the parasite stage that mosquitoes pick up from humans.
Realistic conditions.
Yes. And in the susceptible L224 mosquitoes, about 80% got infected. But in the resistant Q224 mosquitoes, the infection rate dropped dramatically down to around 30%.
Wow, that's a big drop. What about the intensity of infection? Did the few that got infected still carry a lot of parasites?
Even more striking there, they measure intensity by counting oocysts, which are parasite stages in the mosquito gut. The median number in susceptible mosquitoes was 3. In resistant ones, it dropped to 0.
0. So most resistant mosquitoes had no parasites developing.
Effectively, yes. And maybe most critically, they looked at sporozoites in the salivary glands. Those are the stages the mosquito actually injects into humans when it bites.
The transmission stage.
Exactly. In the susceptible mosquitoes, they found thousands, 4,000 or 5,000 sporozoites. In the resistant Q224 mosquitoes, on average, 0.
0 sporozoites. That basically means they couldn't transmit the disease, right?
A near-complete blockage of transmission potential at that crucial final stage. It's incredibly promising.
Those numbers are really impressive. Do they know how this resistance works? What's the mechanism?
Yeah, the study suggests that this protective Q2224 version of the fibrinogen-related protein 1 works really early on. It seems to inhibit the parasite development before the oocyst stage even forms.
So it stops the parasite right at the beginning before it gets established in the mosquito.
Exactly. Blocking it before it can really get a foothold.
Yeah.
And this wasn't just specific to the human parasite.
Oh.
They also tested it against Plasmodium burgei, which infects rodents. And the Q224 allele also significantly reduced infection by that parasite too.
So it suggests a broad spectrum effect, not just tailored to one parasite species.
It points towards a potentially more universal mechanism of blocking Plasmodium development in the mosquito, which is very encouraging.
Okay, but back to that crucial point you raised earlier, fitness costs. Did making mosquitoes resistant make them weaker in other ways? Was there a trade-off?
This is maybe the most important finding that sets this approach apart. The researchers found negligible fitness costs associated with resistant Q224 allele.
Negligible, so no real downside for the mosquito.
Pretty much. They compared things like body size, how many eggs females laid, how long they lived, survival rates during development, and the Q2224 mosquitoes were basically indistinguishable from the normal susceptible ones.
That's huge, because it means these resistant mosquitoes can actually compete and thrive in the wild.
Absolutely vital. If they weren't fit, the resistance trait wouldn't spread effectively, no matter how good it was at blocking parasites. Fitness neutrality is a game changer here.
Okay, so the resistance works, and the mosquitoes are fit. How well did that allelic drive system actually spread the trait in their tests?
It proved highly efficient. They ran experiments in laboratory cages, starting with a mix of mosquitoes where only 25% carried the resistant Q2224 allele.
Right, a minority.
Within just 10 generations, the frequency of that protective Q2224 allele shot up to over 90% in the population.
10 generations, that's fast.
Very fast in mosquito terms. It really indicates the potential for this system to spread rapidly and have a widespread impact if deployed in targeted wild populations.
Wow. And I assume other scientists are taking notice. Has there been external validation?
Yes, definitely. We saw comments from Mosquito, who's an expert in this field but wasn't involved in this study. He called it a, quote, promising path toward modifying wild mosquito populations.
High praise.
And he specifically highlighted its potential as a, quote, cost-effective intervention, which, as you know, is incredibly important and something he said is urgently needed.
That external validation really adds weight. So let's pull back to the big picture. What does this mean for the fight against malaria overall?
Well, it's crucial to see this not as the solution, not a silver bullet that replaces everything else.
Right, not a magic fix.
No, but as a potentially powerful, very sophisticated new tool to add to our arsenal. Think about using it alongside existing methods.
Like combining it with bed nets and sprays.
Exactly. Imagine that multi-pronged approach. You have nets and sprays, reducing human-mosquito contact, and then the mosquitoes that do bite are potentially unable to even transmit the parasite because of this genetic modification. It creates multiple layers of defense.
Attacking the problem from different angles, that sounds much more robust.
It really does, adding a layer of protection within the vector itself, making the mosquito a dead end for the parasite.
And could this be used beyond this specific mosquito, Anopheles stevensii?
The potential is certainly there. The hope is that this strategy could be adapted for other major malaria vectors, like Anopheles gambii, which is the key vector in many parts of Africa.
That would be huge.
And there's even potential, looking further out, to use similar allelic drive systems for other goals. Like, imagine converting insecticide resistance alleles back to susceptible ones.
Wow, reversing insecticide resistance. That would be incredible, turning back the clock on that problem.
It opens up some really interesting possibilities for managing vector populations in new ways.
This deep dive has really illuminated how precise genetic engineering, done thoughtfully, could offer these incredible solutions for huge global health issues. And the self-eliminating aspect is fascinating.
It addresses a key barrier, doesn't it?
It does. It makes you think, doesn't it? It raises questions about the future of genetic tools in public health. How do we balance this amazing potential, this power, with the need for, careful, responsible consideration before deployment?
That's the crucial question moving forward. Balancing the promise with precaution.
Definitely something for all of us, and especially policymakers and communities, to ponder as these technologies continue to develop. It's a powerful example of science offering targeted, maybe even elegant solutions to persistent challenges.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.